Number 7, CRISPR, genome editing and cancer. Being able to read, study, compare, and edit DNA sequences for humans, plants, and animals has always been the focus of researchers around the globe. CRISPR, or clustered regularly interspaced short palindromic repeats, a revolutionary new technology in the research arena, is sweeping the world with excitement due to the unprecedented ease and speed with which it allows scientists to change the genetic code of DNA. Some say CRISPR gene editing may prove to be one of the most important biologic tools invented in the past 100 years. The technology holds great promise for discovering therapeutic targets in cancer and other diseases. This genetic engineering technique allows scientists to manipulate specific genes within cells, turning them off or changing their function. From replacement gene editing to targeting of immune cells against cancer, the technology is enabling an entire new world of possibilities for cancer treatment. Human trials for a rare eye disorder using CRISPR-based gene therapy may begin as early as 2017, and the hope is that this technology can ultimately be used to tackle a wide variety of genetic disorders.
And our expert in this field is Dr. Keith Joung, who's a professor of pathology at Mass General and has been a leading authority in the field of targeting DNA from zinc fingers to CRISPR for a long, long time. So Keith, the question is how CRISPR CRISPRs, and should we worry about off-target effects?
Well, thank you, Daniel. So as the video just showed you, CRISPR is a very exciting technology. It gives us the ability in essentially any type of cell to make any type of change at any locus. So the possibilities for research applications, for doing diagnostics and doing certainly therapeutics is very, very exciting. But as you know, it's also known that even though we've gotten very good at hitting where we want to hit and making a change in a cell, these technologies can also cause changes at other places in the cell where we don't want those changes to occur, the so-called off-target effects. And if you think about the problem, it's really one it's a very difficult and challenging one because many of these therapeutic approaches, you're envisioning modifying tens of millions, perhaps hundreds of millions of cells or more, each of which has a genome composed of 3 billion base pairs or more. And so trying to make sure that you only really hit one place in each cell and nowhere else is a very, very difficult challenge. Now, having said that, does that mean that we shouldn't proceed with these therapies? And I would say the answer is no, because we have the opportunity with these new technologies to be able to develop treatments for diseases for which there really haven't been treatments before. We also have the opportunity to do things like modify cell-based therapies and make them more efficient and safer. The issue really is one, I think, of patient safety overall, of which quantifying these off-target effects is just one aspect of that. And so I think if you think about the problem of going in front of regulators or going in front of patient advocacy groups and making the case for using these technologies to be able to do treatments, part of what you want to be able to do is quantify that risk. You want to be able to say, to the best of your ability, where, how often are these alterations occurring. And the technology is advancing. We're certainly better at doing that than we were 2 years ago. But we still can continue to make forward progress. And again, I think we owe it to the patients to do the best job we can. And that's a technology that will evolve over time. And as it does, then we'll be able to use that as a yardstick to measure as to make improvements to the technology and further reduce the number of off-targets. So I'd say in summary, a very exciting time, very exciting technologies to develop new classes of therapeutics. But we do need to continue to try to do a better job of figuring out where off-targets occur as part of an overall safety evaluation.